The cell cycle consists of several phases, one of which is interphase. Interphase is the longest phase of the cell cycle and is divided into three subphases: G1 phase, S phase, and G2 phase. G1 phase, gap 1 phase. During this phase, the cell grows in size, synthesizes proteins, and carries out its normal metabolic activities. It prepares for DNA replication. S phase, synthesis phase. In this phase, DNA replication occurs. The cell duplicates its genetic material, ensuring that each daughter cell receives an identical copy of the genome. G2 phase, gap 2 phase. After DNA replication, the cell enters G2 phase. During this phase, the cell continues to grow, synthesizes additional proteins, and prepares for cell division. Checkpoints in the cell cycle are critical control points that ensure the integrity of the DNA and the proper progression of the cell cycle. There are three main checkpoints. G1 checkpoint, also known as the restriction point. This checkpoint occurs near the end of the G1 phase. It ensures that the cell has the necessary resources, growth factors, and appropriate conditions for cell division. If the conditions are favorable, the cell proceeds to S phase. If not, the cell may enter a non-dividing state called G0 phase or undergo apoptosis, programmed cell death. G2 checkpoint. This checkpoint occurs at the end of the G2 phase. It checks for DNA damage, verifies the completion of DNA replication, and ensures that the cell is ready for mitosis. If any abnormalities or DNA damage are detected, the cell cycle may pause for repairs or activate cell death pathways. M checkpoint, also known as the spindle checkpoint. This checkpoint takes place during metaphase, the stage of mitosis where the chromosomes align at the cell's equator. It ensures that all chromosomes are correctly attached to the spindle fibers before the cell proceeds to anaphase, where the chromosomes separate and move to opposite poles. If any errors or improper attachments are detected, the checkpoint halts the progression of the cell cycle until the issues are resolved. These checkpoints are crucial for maintaining genomic stability, preventing the proliferation of damaged or abnormal cells, and ensuring proper cell division.